Hi class, welcome to the online session. Our topic for today would be on the central nervous system, and we are going to take on the following slides. We have the cerebellum, the cerebrum, the spinal cord, the spinal ganglion, the nerve in cross section and longitudinal sections, the neurovascular bundle, the motor end plate, the sympathetic nerve ganglion, which is similar to the spinal ganglion. We have the optic nerve, Pacinian corpuscle, and the Meissner's corpuscle. Okay, so let's start with the first slide is the cerebellum, okay, a cerebrum. So for the cerebrum, let us try to look at the entire slide. So the cerebrum would be divided into two regions. The outermost region, which is this portion, this one, okay, up to this area, okay, this would be called the gray matter, okay. And the central portion of the cerebrum, you can see the presence of these horizontal layers of fibers. This would be called the white matter, okay. Here again, you can see the horizontal layering of the fibers, and that would constitute here the white matter. Okay, so now let's go into the gray matter. Okay. So the gray matter would be composed of several layers, but in some areas, it is very difficult to distinguish one layer from the other. So the one that is very prominent, okay, among the six layers of the gray matter would be called the plexiform or molecular layer. And this is the outermost layer, which is composed mainly of fibers. Okay, so if you're going to look at it at a higher magnification, like this, you can see that we would only appreciate a few neuroglial cells, and then you have a lot of those fibers. Okay, so when you say neuroglial cells, this refers to the supporting cells, which are composed of the astrocytes, the oligodendrocytes, and the microglial cells. Okay, so I think these are mainly composed of astrocytes. Okay, so if we're going to go to the next layer, it is called the small pyramidal. Okay, so we have the small pyramidal. This would be the layer. This would be followed by the external pyramidal. By the way, the small pyramidal has several uh, names. Okay, we have the external or outer granular layer. Okay, and then we have the third layer. It's called the external pyramidal or the medium pyramidal. And then we have the fourth layer, which is the granular or internal or inner granular layer. And then we have the ganglionic. Okay, so where is the ending? So you have the ganglionic, and then the last portion, which you always would situate nearest to the white matter, would be the polyform or multiform layer. Okay, so it's very difficult to separate them or distinguish them. The only thing that you would be able to separate would be the molecular layer, the plexiform, and the one that is situated nearest or closest to the white matter, which is called the multiform or polymorphic layer. So in between, you may have some overlapping boundaries. What would be prominent in the second to the sixth layers would be the presence of these cells, okay, here, which are called the pyramidal cells because of their appearance. Okay, they are triangular there. Okay, and these are neurons or neuron cell bodies. Okay, the other cells present would be composed of granule cells, which may be difficult to differentiate from the astrocytes. Okay? So the inner portion, this one, okay, this one. So you can see the presence of the horizontal layers of the axons. So this would now be the white matter. Okay, so the white matter would be composed mainly of myelinated and unmyelinated fibers, and you can see the presence of neuroglial cells in the area. So there's absence of neurons in this portion. Uh, you can see the presence of cells having moderate cytoplasm here, and these are oligodendrocytes, which would be the producers of myelin in the central nervous system. For the others, you have the ones with scanty cytoplasm, they are the astrocytes. Okay, so if you'd be able to see a smaller cell that would possess elongated nuclei, that would be the microglial cell. Okay, so now we go to the cerebellum. Okay, so cerebellum, let's look around the tissue or the slide. Okay, more or less, we have similar appearances. Okay, if you're going to look at this area, this would be the gray matter from this portion up to here, and that's the outer portion. The inner portion would be the white matter. Okay, so it has the same appearance as that of the white matter of the cerebellum, of the cerebrum. So let's look at the high power magnification of the area. So this would be the gray matter, and there are two layers to it. So the outer layer would be the molecular layer, and if you're going to look at it at a high power magnification, it would be composed mainly of a few neuron cells, which are called the basket cells, and then you have the nerve fibers in between. And then in between, or found in the boundary between the molecular layer and the granular layer, would be large cells, okay, which are called the Purkinje cells. So if we're going to look at the low power magnification, so you can see large cells set at the boundary. So these are Purkinje cells. So what are Purkinje cells? Purkinje cells are large cells. They look like ganglion cells, as they possess a large cell body, prominent nucleus with prominence of the nucleolus, and then you see the presence of this um, of these dendrites. Okay. Okay. So and then the innermost layer of the gray matter would be called the granular layer, as it is composed of small neurons, which are called granule cells. Okay, there. So they look like lymphocytes. Okay. So, and then if we're going to look at the, wait, 'cause- okay, it's okay. So if we're going to look at the innermost portion, you can see the presence of the white matter. So this would be the white matter. So this is the white matter, okay, and it would be composed mainly of, again, the neurons, and the neuroglial cells. Okay, so now let's go to the spinal cord. Okay. So for the spinal cord, let's move it around so that you would appreciate it. So this is a big tissue. Okay, so let's start. So the outer surface of the spinal cord, we have this connective tissues that would surround the spinal cord, which we would call as the meninges. There are three meninges that surrounds the spinal cord. The outermost, this would be the dura mater, and this is the thickest among the three. And the next would be we have the arachnoid. Okay, this is the arachnoid, and then this is the pia. Okay, let's look at the high power magnification of the three. Okay, so with regards to the spaces in between them here, this is the space that is lying underneath the dura. We call it as the subdural. Okay, the subdura would be composed of the venous sinuses. And then underneath the arachnoid, we have the subarachnoid space. This is subdural space, sorry. And then you have the subarachnoid space. In the subarachnoid space, it would contain the cerebral fluid, okay, cerebrospinal fluid, which is the CSF. And also transecting within the subarachnoid space are these uh, arteries. Okay, so that's where we would be able to see the branches of the cerebral artery. Okay, now we go to the inner portion. So the within the spinal cord, you would see the presence of a whitish portion. Okay, and this is called the white matter. The white matter would be composed of axons there. So you can see the presence of the axons. The one in the center there. Okay. So the axon here would be this one, and it's surrounded by the myelin sheath. Axon, myelin sheath. And then, of course, you have the endoneurium. Okay. So if we're going to look at the central portion, so this is the central canal, and then the connecting sheath would be called the gray commissure. The gray matter would be this butterfly shaped area. Okay, and the one on top would be the dorsal. The one at the bottom would be the ventral, and the one at the bottom would possess the large motor neurons. Okay, so you can see the large motor neurons. Okay, so this large motor neurons, you would be able to see the presence of the dendrites. This is the cell body. You would see the large nucleus, and then at the center you have the nucleolus. Okay. And there are processes that would be present, okay, that would project from the cell body, and these are either the axon or the dendrite. Within the cytoplasm, you can see the presence of this. these colored structures, which are actually the missile bodies. There, let's try to see this one. Okay, so you can see the presence of those colored structures, which are called the missile bodies. The missile bodies are actually the rough endoplasmic reticulum that would contain the ribosomes. So how to differentiate an axon from a dendrite? If you do not see missile bodies, okay, into the process, then it's an axon, which is this case. If you see the presence of uh, the missile bodies going into the process, then that's a dendrite. So the action of the axon is that it will transmit impulses from the cell body to other cell bodies, while the dendrites would receive impulses from other cell bodies to the cell body. Okay? And then you would see the presence of smaller cells, which are the central neuroglial cells or neuroglial cells. Now let's go to the nerve. So in this case, we have two nerve fibers. What is important here is you would be able to identify the connective tissue that covers the nerve fiber. So the this is a nerve fiber. Uh, there's the dense irregular connective tissue that would surround the entire nerve fiber. It's called epineurium. Okay. Uh, in this particular nerve fiber, it's very difficult to see or identify the perineurium. So let's try to identify the endoneurium. So the endoneurium would be the it's a loose connective tissue that would surround individual nerve fiber, nerve, yeah, uh, nerve fiber. Okay, so okay, so the I'm sorry, the endoneurium would be for the nerve fiber. For the entire nerve, we are not going to call it as a nerve fiber. The entire nerve or the peripheral nerve would be for the epineurium. So the one in the middle of the nerve is the axon. Okay, and surrounding it would be a clear substance, which would be the myelin. Okay, myelin sheath or myelin. And then you have the endoneurium, which is a loose connective tissue. Let's try to see the optic nerve. And in this case, you would be able to appreciate the presence of the perineurium. So again, for the optic nerve, the peripheral nerve is surrounded by a dense irregular connective tissue, which is called the epineurium. Okay, this one. And then this time, you would be able to identify for some nerve fascicles. Okay, here. So you have the nerve fascicle over here, which would be surrounded by this specialized connective tissue, which would be called the perineurium. So this perineurium is special because it is a blood nerve barrier, a blood nerve barrier. And then, of course, you would see the presence of individual cells, individual axons surrounded by the myelin, and then you have the endoneurium. Now let's look at the nerve in a longitudinal fashion, okay? Because the one that we saw would be the cross section. So here you would see the horizontal fibers. Okay? So you need to appreciate the the fibers. You would see the central portion, this one, that would be the axon, surrounded by a clear substance, which is the myelin, and surrounded by the endoneurium. So some cells are present here, which are fibroblasts, and then you have the round cells, which would be the Schwann cells, although sometimes it's very difficult to separate them. And you can see this portion, okay? This is the junction between the axons that are the junctions of of unmyelination, and this would be called the node of Ranvier. Okay, the node of Ranvier. So if we're, let's try to look at the other areas. So again, the one in the middle, okay, this is the axon. Okay, that's the axon. And the neurium, axon. Let's look at some of the other areas. Sometimes here. So this would be the node of Ranvier. Okay, so is it myelinated or unmyelinated? It's the unmyelinated portion. Okay, here, it's another one, the node of Ranvier. Okay, so next we go to the ganglion. So the ganglion or the spinal ganglion will be located in the peripheral nerve there. Okay, and this time we can see the presence of large cells there. So these large cells, okay, so they would possess large nucleus, large cell body, prominence of the nucleolus. They are surrounded by nerve fibers and these round cells, which are called the satellite cells. Okay, so notice that there's a little bit of granularity in the cytoplasm, and that's because the cytoplasm of the ganglion cells would contain lipofuscin pigments. Let's try to look at the longitudinal section of the spinal ganglion. Okay, and this one would show better view. Okay, so you have a lot of those ganglion cells, prominence of the nucleolus. The they would possess pale nucleus. Okay, you see a lot of those lipofuscin pigments there in the cytoplasm, and then you have the presence of the satellite cells. Nerve fibers are present uh, in a longitudinal fashion. Okay. Now we go to the, so I think this would be the neurovascular bundles. Okay. So for the neurovascular bundles, it simply would show to us the presence of the artery. The this would be the vein, and you can see the presence of this peripheral nerves or nerves. Again, what we see, presence of the epineurium. This would be the endoneurium, perineurium, sorry. And then you have the endoneurium. Epi, you have the peri, and then you have the endoneurium, which is a loose connective tissue, specialized connective tissue, dense irregular connective tissue, if you're going to be asked the type. Next, we go to the Pacinian corpuscle. Okay, take note of the spelling. So for the Pacinian corpuscle, it can be situated in the skin. It can be situated in the deeper portions, like in this organ. This is already a pancreatic tissue. This is a Pacinian corpuscle. And a Pacinian corpuscle is a sensory receptor. It is a sensory receptor for pressure and vibration sense. It is composed mainly of flattened Schwann cells there. So it's composed mainly of flattened Schwann cells or fibroblast-like cells found in a lamellar fashion. Okay. Then we have the Meissner's corpuscle. So what do you think this tissue would be? So this is the skin. You have the epidermis, the dermis, or the stratified squamous epithelium. So we are going to look for something in the dermal papillae. When you say dermal papillae, that is at, underneath the basal portion of the epidermis. Okay? So what are we going to look for? Okay? So we are going to look for something like this one. Okay, so it's like a bullet there. So we are going to look for these aggregates of Schwann cells, okay, composed of flattened Schwann cells, and this is a receptor, sensory receptor for touch. Okay, so I think that's the only one that I saw. Mhm. Okay, so our last slide for this session would be the motor end plate. Okay, so what we are going to have here is a gold chloride stain. This would be the skeletal muscle fiber. And what we would see here would be the terminal branches of the axon. Uh, and this would be the junction between the axon and the skeletal muscle fiber. This would be called the motor end plate or the neuromuscular junction. Okay, so that ends our session on central nervous system. Kindly read your book, okay, for review and study of the chapter. Thank you and good day.